Hello, my name is Amber Schmidtke, and this will be a lecture on the circulatory system. It is for class Biology 1001 at Middle Georgia State University. This is a class for biology non-majors, so it is just sort of a quick survey of how the circulatory system works. The learning objectives for this, come on now. The learning objectives begin with, we want to make sure that we talk about the commonalities that you see across all circulatory systems. Then we will learn in detail about the human circulatory system, and including what happens when things go wrong, like a heart attack or a stroke, that sort of thing. And then we will identify the components of blood. We will also spend some time talking about the interaction, once again, between the respiratory system and the circulatory system, because they really do go hand in hand. Now, circulatory systems are something that's necessary for any animal whose body is too large for exchange through diffusion alone. As you can probably appreciate, we are a very complex organism, and so we do have to have a specific system for carrying things like nutrients, gases, and waste. Circulatory systems are a network that reach nearly every cell in the body to deliver nutrients, gases, and again, to dispose of that waste. And all of them really have three critical components. First is they have a muscular pump known as a heart. The second thing is that they have blood that will be transported through blood vessels and the heart. So that circulatory fluid is blood, and of course the vessels carry that blood or fluid from the heart to the tissues. And an important thing to note is that we do this thing where we have what's called a closed system, where the blood and the interstitial fluid are separated from one another using the blood vessel walls. And so interstitial fluid just refers to the fluid that bathes the animal cells themselves. It is a distinct sort of fluid from blood. It's mostly water. And nutrients and gases will travel through the space, using simple diffusion. So, for example, this is a simplified sort of drawing of maybe an animal structure. And of course, we've got the digestive system over here on the left. We've got urinary and respiratory, but what we're mainly paying attention to is the circulatory system, reminding ourselves that the circulatory system's main role is the transportation of that blood, which carries, again, the nutrients, the gases, and the wastes. So, the heart, of course, pumps blood throughout the body, but the blood has to get those nutrients, gases, etc., to the tissues themselves. And so what will happen is that those items will diffuse through the wall of the blood vessel into this interstitial fluid and then migrate to the body's cells. And then wastes and carbon dioxide and those sorts of things can travel back into the bloodstream, heading to the heart to be, or the kidneys to be expelled. So there are two different kinds of the circulatory system. You can have an open system where the blood and the interstitial fluid mix, and you really can't distinguish them from one another. And then you can have a closed system, which is what we have, in which case the blood and the interstitial fluid are kept separate from one another, and it's really kind of serves like a membrane like you see in osmosis, where things will move from one area to the next, but they don't actually touch. So in an open circulatory system, this is something you would see in many invertebrates or animals that don't have spinal vertebrae. So this could include a grasshopper like you see here, mollusks, and all other arthropods or insects, or mostly insects. And so what they use instead of a heart in a circulatory system like we have is they will have a heart pump too. And what happens here is the body fluid will go from that tubular heart, like you see here, to the head and then to the rest of the body through these sort of vessels. And there it's going to mix directly with the interstitial fluid. The fluid is then pooled and returned back to the heart when the heart relaxes. And then, of course, this is going to interact with the gases in the tracheal system, which is their breathing apparatus. In a closed circulatory system, this is what you see in vertebrates like us, something that has a spinal cord, an earthworm, squid, and octopi, for example. And so in this situation, the blood is confined to blood vessels, and therefore it is separated from the interstitial fluid. So we have three different kinds of vessels that we want to talk about. We have arteries, and I always think of A as away because it carries blood away from the heart. And then we will have veins, which are always going to be returning blood to the heart. So they are heading towards the heart. The arteries are heading away from the heart. Capillaries are sort of this intermediate that can exist between arteries and veins, where blood is transported through very, very tiny blood vessel networks to facilitate their movement into the tissues, or to facilitate the movement of gases and nutrients into the tissues and out. And that's sort of the pivot point between when a vessel transitions from being an artery to being a vein. Because once those capillaries collect the waste products, collect the carbon dioxide from those tissues, then those resources need to be pooled, and then returned to the heart to be carried out to either the lungs for the carbon dioxide or the kidneys for the waste products. So in our situation, most often our closed circulatory system is also going to be referred to as a cardiovascular system. And so we'll talk about this first in the context, we're looking at this fish for an example, but a heart is going to have two main chambers. There is going to be an atrium or atria in plural, and this is what's going to receive the blood from the veins. So it's sort of the holding tank temporarily for the blood that is coming back to the heart. Now the blood can be coming to the heart from the surrounding tissues, or it could be coming directly from the lungs. So there's two atria that we're going to need to talk about. And if it helps you to remember, the main entrance to our neighborhood, or not neighborhood, our building has that great big vaulted ceiling, and you will sometimes hear that referred to as an atrium. So if it helps you to remember that an atrium means a tall ceiling, the atria are the top part of the heart. So hopefully that helps. The ventricle is the bottom part of the heart, and you have two chambers for this in our system too. And so what these do is they pump the blood out of the heart. So blood comes in through the atrium, it leaves the heart through the ventricles to transport throughout the body, to transport through the body. So if we look at this in a fish, what's going to happen here is we've got the heart here. And remember that in our graphics, we tend to depict oxygen poor blood in blue, oxygen rich blood in red, but it's important to realize that that is not how it works in an actual living thing. Your blood is not blue. We just use this as sort of a trick to keep you aware of what's going on. So in this situation, oxygen poor blood is going to leave the heart and head to the gills in this fish. And because of countercurrent exchange, which we talked about last time during gas exchange, the oxygen is going to transition from being oxygen poor across these capillaries to oxygen rich. Those oxygen rich blood cells are going to then fill into these arteries, which are going to then transport to different parts of the body of that fish, the capillary beds, where they can feed the tissues. And then blood will return back to the heart. So we have, just like we saw in the lungs, you could have bronchi that then narrow into bronchioles. The same sort of thing can happen with these blood vessels. And so we have large arteries. These are, again, the vessels that carry blood away from the heart. They can branch into smaller vessels called arterioles. And those will eventually, that's what we're seeing here, is a narrower version of an artery, but they will eventually bridge into these capillaries. And capillaries are very, very thin networks of blood vessels. They're very, intricate and convoluted, like you see here. And what they do is they deliver blood to the outlying tissues. And so you've got these that support all sorts of different parts of your body. There are capillaries that surround the alveoli in the lungs. You'll have them embedded in different parts of your body, that sort of thing. There are sphincters that control the flow of fluid through here. And one of the major responsibilities here is to prevent backflow. So we want blood to flow through this, but not necessarily backward. And so we have these sphincters that help to control that movement. But capillaries themselves, or capillary beds, have very, very thin walls, not like what you see in an arteriole or a venule, which we'll talk about next. So this means that there's going to be easier diffusion between the bloodstream and the interstitial fluid. Eventually, these capillaries will then funnel into what's called a venule, which is the counterpart to the arteriole, But where this blood was being carried away from the heart, now it's time to transition to pooling these waste products into a vein, or an early vein called a venule, which will lead back to the heart for disposal. So the venule is really the exit point for the capillary bed. In humans, we do what's called double circulation, which means that because we have two atria, because we have two ventricles, there's going to be two separate circulatory paths. And so you will have a situation where your blood pressure will drop when it arrives at the lungs, and there will be a second pump through the heart that then helps to propel the blood through the body. So you have what's called the pulmonary circuit, which carries blood away from the heart to the lungs and then back to the heart in order to exchange gas with the external environment. Then you have the systemic circuit, which is going to carry blood between the heart and the rest of the body. Now that blood can be oxygen rich if it was picked up by the lungs or from the lungs, returned to the heart, and then left the heart to go to the tissues. Or it could be talking about the oxygen poor blood that's on its way back to the heart. So the heart has two atria and two ventricles for a total of 4 chambers. The right side of your body, or the right side of your heart, handles the oxygen poor blood. So this will be the blood that is heading to the lungs and will be and will therefore enter the pulmonary circuit. The left side of your heart handles oxygen rich blood. So this would be the blood that comes directly from the pulmonary circuit, and now we need to send that out to the body tissues. So that is called the systemic circuit. All right, so we will then keep going here. Oh, and one more thing I wanted to mention. When we talk about the right side, left side, keep in mind that that's from the perspective of the organism that owns that organ. So for example, when you're facing me, my right ventricle or my right atria will look like it's on the left-hand side to you, but it's always from the perspective of the person who owns the organ, or the organism that owns the organ. So here's a picture that helps us to better understand how the pulmonary and systemic circuits work. So here is the heart. These upper chambers, what's labeled here as a green number 4 and a yellow number 5, those are the atria. Again, because everything's going to look opposite, this is the left ventricle, and this will be or left atria rather, and this is the right atria, or atrium, excuse me. Down below you have the ventricles. They're bigger in size than the atrium, or atria, excuse me, I keep getting these mixed up. And so the pulmonary circuit is going to include all of these veins and arteries that go straight to the lungs and back. So there will be pulmonary arteries. Again, A means away. So they're going to carry oxygen poor blood to the lungs. So they're shown here in blue because they are oxygen poor. They have to go to the capillary beds that surround the alveoli inside the lungs to pick up fresh oxygen and to expel that CO2. Once that oxygen-rich blood is pooled back into the venules, they will fill into the pulmonary vein, which returns blood from both lungs, there's one on each side, to this atrium right here, the left atrium, which is labeled number 5. From here, blood will move from the right atrium, or excuse me, the left atrium into the left ventricle, and then it will leave the heart to start the systemic circuit via the aorta, which is an artery, but it's given this special name, the aorta, because it is probably the biggest deal of all your arteries. So this aorta feeds all of the cells of your body. So this aorta is this blood vessel. It leads to the capillaries of the arms, chest, and head, but it will also feed the bottom parts of your body too, including the abdominal region and the legs. So it'll split into those two paths. Then when the blood needs to be pooled from the capillaries and returned to the heart, it's going to return using veins. We have what's called vena cava. And that's because they're the main vein returning back to the heart. You have what's called a superior vena cava, which covers the top part of your body. It is not called superior because it is better than the inferior vena cava. It is called that because it refers to the top part of your body. You also have an inferior vena cava, which returns blood from these capillaries in the lower extremities and in the abdominal cavity back to the heart, where it will reach the right atrium And remember, the oxygen, it's very oxygen poor blood at this point, so it's labeled in blue. It will pool here in this atrium, go down into the ventricle, and then resume the pulmonary circuit heading to the lungs. So the human heart is mostly going to be made of cardiac muscle, but there's also going to, as we talked about when we did chapter 20 and we talked about the different tissues and the muscles and the nervous tissue, for example, it's actually comprised of not just cardiac muscle, but it's also comprised of things like nervous tissue. It's because nerve impulses are what drives the heartbeat. There's also going to be connective tissue there in the form of blood. There's epithelial tissue for the blood vessels, that sort of thing. And so what you'll notice, if we look at this a little closer, is that the walls of the atrium are relatively thin, but the walls of the ventricle are much thicker. And that is because they have to exert a lot of force to move that blood out of the heart to where it needs to go. And what you'll notice is that the left ventricle has an even thicker wall to it than the right ventricle does, because this blood that's pooled here in the right ventricle only has to get to the lungs. It does not have to get to the surrounding parts of the body. So the blood in this part of the heart has a much further distance to travel. So we need more force to expel this from this compartment, leading to the aorta and distributing it through the tissues. All right, so that is the structure of the heart. So we use a series of valves that you'll notice here, and what they do is they provide the ability for unidirectional blood flow. We only want it to go in one direction with very little, if any, backflow into the previous chamber. So if blood is coming into this right atrium, it'll initially be held here because this valve here called the atrioventricle valve or AV valve will be closed. But when the heart beats, what's going to happen is blood will flow from this space into this one, and the valve will close behind it, trapping the blood in here, waiting for the sort of the second part of the heartbeat. Because remember, it goes sort of ka-thunk, ka-thunk. Once it's held in here, that second part of the ka-thunk will then push it through this semilunar valve towards the pulmonary artery. Leading to the lungs. And so you've got a semilunar valve for each ventricle, and you've got an AV valve for each atria leading into the ventricles as well. All right, so let's talk about, I mentioned heartbeat, let's talk about the heartbeat. So in the cardiac cycle, we've got some things that are happening here. When the heart contracts, it's doing that because it's pumping blood. And when the heart relaxes, what's happening during that time in between heartbeats is it's pooling blood in its chambers. So when we have a heart that is relaxed, we are in what's called diastole. And during this time, the blood is flowing into all four chambers. All of the valves or the AV valves are open, but the semilunar valves are closed, keeping the blood inside the heart. During this time, the ventricles nearly fill with blood. Then we transition into what's called systole, and this is the heart contraction phase. So during this time, the atria contract to make sure that all of that blood is going to completely fill these ventricles. Then you have the AV valves are going to close, preventing any backflow back into those atria, and the ventricles are then going to contract. The semilunar valves will open during that process, and the blood will be pushed into the arteries leading from the heart. During this contraction of the ventricles, the atria relax during this time so that they can pool blood for the next contraction or diastole. So the left we mentioned this a couple slides ago, but I hope you noticed again that the left ventricle is going to have thicker walls than the left, or the right, excuse me. And that's again because it has to transport that blood a further distance throughout the body. So we need to be able to provide more reinforcement for that important movement. So it's important to note though that although there is that thicker wall there, both ventricles are actually pumping the same volume of blood. We quantify that using a term called cardiac output, and that describes the volume of blood that each ventricle is pumping per minute. So now that we know a little bit about how the heart contracts, let's talk about how that cardiac cycle is controlled. The heartbeat is an autonomic function, meaning that you do not have control over it. You can't think hard and stop your heart from beating. This is something that happens with or without your notice. It still beats while you're sleeping, and again, you don't have any control over this. So it does not require a direct signal from the nervous system, although there is nervous tissue there that's driving this. So, or at least it doesn't require a signal from your conscious nervous system is what I should say. So what we have here is we have the sinoatrial node, also known as the SA node. This is a nerve or a group of cells that function as the pacemaker. They set the rate of the heart contractions. You'll notice it's found in the upper wall of the right atrium on that side of the body, and it generates the electrical impulses that drive that heartbeat contraction. So when the SA node signal spreads to both of the atria, then they're going to contract in unison. And so then we'll have the impulse relay from the SA node to another node. That's called the AV node, which stands for atrioventricular, because it's that junction between atrium and ventricle. This is located between the right atrium and the right ventricle. And at this time, specialized muscle cells are going to relay the signals to the apex of the heart, which is down here at the bottom. And then up through the walls of those ventricles, you're going to have this impulse to trigger strong contractions that will drive the blood out of the heart. So these are those specialized muscle fibers that are featured in cardiac muscle. So we have talked about now how the heart works, how the circulatory system works. But it's important to take a moment to learn about what we know about the heart in many ways is because we've learned about what happens when things go wrong, as is often the case in science. So you can have what's called an arrhythmia. And this is something where your heartbeat is not functioning at a normal rhythm. And so this is something that's usually detected through things like electrocardiograms, which can be abbreviated as ECG or EKGs. It can either be a situation where your heartbeat is too fast or it can be too slow. And so this can lead to fibrillations or flutterings that you sense in your chest. And these flutterings that take place in the atria or ventricles can occur in healthy hearts. In fact, it isn't necessarily a sign of heart disease when a person is overstimulated by drugs or caffeine use. So this is something to be especially aware of as a college student, because I understand you're under a lot of stress and maybe relying a lot on caffeine to get you through, especially things like finals. So if you start to notice heart arrhythmias where it sort of just feels like your heart pauses for a second and then resumes, then try to cut back on the caffeine, see a doctor, that sort of thing. We can install an artificial pacemaker if a person is having severe problems with a heart arrhythmia and cannot maintain the normal rhythm. That is a surgical procedure that we can do. Another problem that can happen is called atherosclerosis. And what happens here is that the arteries are gradually impaired because of chronic cardiovascular disease. You will have fatty deposits called plaques that line the inner walls of the arteries. And when they do that, they sort of obstruct the flow of blood because they narrow the path in which that blood can travel. So what this can do, because those plaques can dislodge or they can get trapped in places too, is it can result in a heart attack or a stroke if blood flow is completely blocked. And so in a heart attack, what's going on here is the SA node that we talked about before when we were talking about heartbeat contractions is unable to maintain that normal rhythm. We use a defibrillator sometimes to apply an electrical shock, and the purpose of that is to really to push the reset button, if you will, on the SA node to get it to start doing its job correctly again. Now remember that the heart is a very active muscle. This is something that has been beating since you were in utero, and it is working every few seconds from that moment until the moment you die. So it is a hard charging sort of muscle tissue. Because of that, it needs a lot of nutrients and oxygen. So if we have a situation where parts of the blood are not getting the nutrients or oxygen that they need, they will start to atrophy or die very, very quickly. And that's called a myocardial infarction, another word for heart attack. This is irreversible damage. So if we look at the heart down here at the apex, you'll see that there was a blockage right here. We don't know fully what this is, if this is because the capillary got so narrow that because of plaques that blood couldn't flow, or maybe a clot got stuck there, that sort of thing. And so what's going on here is these tissues didn't have the nutrients or oxygen when they needed it, so they died. The problem, though, is that because cardiac tissue is one of those sort of terminally developed cells, they stop dividing once they get to terminal development. And they kind of just stay there in perpetuity. So if you have a part of your heart die like this, we cannot replenish that. We cannot regenerate like we can your liver or other parts of your skin, that sort of thing. That's why you can have these long-term problems after a heart attack, that you're kind of at this weakened state, is because part of your heart is not working and is not expected to recover. All right, so that's a heart attack. A stroke is when you have this situation, but it's taking place in the brain because of a lack of oxygen, or maybe there's a rupture or blockage of the blood vessels that exist in the head. So you can get either one of those things. Blood vessels, we want to take another closer look at that. Now, earlier we were talking about arterioles and how they feed into capillary beds and then they funnel into venules, et cetera, et cetera. But I want to look a little bit more at the cross section of the artery versus the vein. Now, all of these blood vessels, regardless of who they are lined with epithelial cells. Those are the cells that we learned about back in chapter 20. They're the ones that are going to be surrounded by smooth muscle in this case to help drive the movement through these vessels. Then there's connective tissues underlying that, and then we have the wall of the artery. So what you'll notice is that the structure is roughly similar between the artery and the vein, but something you'll notice right away is that the same stuff is there, but it's in different proportions. So where over here the epithelium of the artery is relatively narrow, and we've got a lot of smooth muscle to support that, over on the vein side, the epithelium is quite a bit wider in diameter. We're going to have a thinner space of smooth muscle. And a lot of this is because the arteries are going to be carrying blood away from the heart to the branches and into the arterioles, or they branch into these arterioles and into the capillary beds. They have these thicker walls because they're having more pressure exerted on them than the veins are, because this blood is still carrying the force of the movement of the heart, the ventricle that pushed it outward. So lots of pressure. The other thing is we've got this outer layer of connective tissue that is very stretchy, and this allows the muscle and the epithelium that line the interior surface of this blood vessel to stretch when blood is coming through, and then recoil or sort of snap back when the blood has passed. The vessels can narrow or widen using smooth muscle, and this is something that is going to be sensitive to either hormones or nervous system signals, like if we need to increase blood flow because of exercise or something like that. Capillaries are, again, these narrower, tiny little blood vessels that feed the tissues. And so they are going to be just a single layer of epithelium surrounded by extracellular matrix. What this does is it makes it really, really easy for nutrients and gases to diffuse across that blood vessel wall. The cells of this capillary, the outermost cells, are going to be surrounded in interstitial fluid, and that's what we're looking down here in this cutout. So here's the capillary wall. It is one cell layer thick of epithelium. Here are the blood cells, and then we've got the interstitial fluid shown in clear fluid out this way. So what's going to happen in this case is diffusion once again. So the gases like oxygen and carbon dioxide are going to be able to move from an area of high concentration to low concentration. So in the blood, especially if this is the artery side of a capillary, there's lots of oxygen present. And there will be less present in these cells because they are using oxygen at full tilt most of the time, because they are needing to use that to break down glucose, and oxygen is that final electron acceptor in the electron transport chain. So they're using it all the time. So oxygen is in low supply in the cell, but it's in great supply here in the capillaries. So we've set up a gradient. And remember from way back when we were talking about cell membranes, how osmosis and diffusion work, that the particles will naturally flow from an area of high concentration to low concentration until they reach equilibrium. Now, the opposite is true for carbon dioxide. Because we are at this junction of the capillary bed, there's not a lot of CO2 there yet. And so we're building up a lot of carbon dioxide here in the cells because they're digesting glucose. And one of the byproducts of that is production of CO2, or release of CO2. So there's concentrated CO2 here, but there's not a lot in the blood vessels. So what happens is we, again, set up that gradient. It's just moving in the opposite direction. And so what happens then is all of this oxygen poor but CO2 rich blood is going to drain into this sort of funnel that's referred to as a venule that leads back to the vein. There we go. So the veins themselves are going to carry blood from these capillaries back to the heart. And what's going to happen here is that because we have that lower blood velocity, like the force of blood from the artery to the capillary bed is really high, but funneling these things takes a lot less force. Because of that, they're going to have thinner walls to their blood vessels. And then, of course, large veins are going to have these flaps called valves. And what they do is they're unidirectional, only allowing blood to move in a particular direction, in fact, forcing it to go back to the heart rather than staying pooled here. And that's important when you think about things like gravity. You can walk around all day, and gravity, if that was the only thing that mattered, would mean that most of your blood is pooled in your feet. But it's not. Because of the force that's exerted by the ventricle through the aorta, that blood is being moved, and then every time it reaches up a higher level, we trap it in that space using these valves. And then with the next smooth muscle contraction, it moves up to the next space. All right, so next let's talk, now that we've talked about when things go wrong, let's talk about blood pressure and how it relates to heart disease. So blood pressure is describing the force that blood exerts against the walls of the blood vessels, those arteries, right after it leaves the heart, when they're driving blood from the heart and through the arteries and capillary beds. So we're just talking about after that muscle contracts, the amount of force that's pushed up against those artery walls. Your pulse is something you can measure, and this actually is the rhythmic stretching and recoil of those arteries to accommodate the overflow of blood. So when your heart beats, a massive amount of blood is going to be pushed immediately into the closest part of the aorta, the nearest and most important artery. When that happens, it's a lot of blood traveling through a narrow space. And so the blood vessel has to expand in order to hold that briefly. And then once the blood passes through, it can recoil back to its original shape. So that is what you are hearing when you take a pulse, or when you feel a person's pulse. That's what you're feeling, is that stretching of the arteries while the blood is waiting to get into the arterioles and into the capillary beds. So if we look at this from a little bit more of a perspective in which we can measure things, you're going to have what's called systolic pressure and diastolic pressure. Because remember that we have the diastole state of the heart, and then we have the systole state of the heart. And so those things, all of this relates back to how the heart contracts. So if we look at this situation here, in the first area, we're looking at the aorta, then we're talking about the arteries, then we're talking about the arterioles, then the capillaries, the venules, the veins, and the vena cava. So that labeling scheme carries on up here too. And so if you're looking at heart heartbeat, excuse me, you'll notice that you have this, and pressure, you'll see that you have this sort of wave pattern of your systolic and diastolic pressure. So when the heart is beating, then the pressure is very, very high. When the heart is relaxing and pooling blood, then it's very, very low. Now, what's this whole graphic here that we're looking at is not just a function of pulse or blood pressure, but it's also talking about blood velocity, which means how fast is the blood moving throughout the body. So what's happening here is we've got really robust pressure, and therefore we have really robust velocity moving through the aorta. This is the first artery, the major artery that leads away from the heart to feed the tissues. It then starts to distribute into the arteries. So what's happening here is it's a force divider. So if this is a force that's being pushed through here, we're then going to divide that force across 3 different arteries. And so it's not going to be as strong as it was before. So what you'll see is this diastolic and systolic pressure start to decline. And we start to see that curve coming down here in terms of velocity too. So the blood flow is slowing down slightly. Let's go back. When you reach the arterioles, we're going to branch that even further. So the pressure is going to drop off, the velocity is going to drop off. In the capillaries, it virtually reaches 0 because we have that interaction between the blood and the interstitial fluid, and so it's hard to measure anything during that time. But then the blood starts to pool into the venules, and so we'll start to see this small increase in blood velocity. Meanwhile, pressure is virtually non-existent on the vein side of the blood circuit as compared to the artery side, because at this point it's weak pressure that's driving things. The venules will gather up that oxygen poor blood, funneling it into veins. And so now we're starting to pick up velocity, because where this was a trickle, we're now combining these into what might feel like a flood inside of your body. And then, of course, these veins all pool into either the superior or inferior vena cava. All right. So I think that pretty much covers that. Did we mix too many? No. So it's important to also understand that, there's a lot of pressure being exerted on the artery side of this. And when we get to the capillary beds, we kind of don't want a lot of velocity here, because we want to have that maximal opportunity for exchange between the blood and the interstitial fluid. But by the time that the blood enters the veins, then it's encountered so much friction from this interaction here in the capillary bed that we're no longer really feeling the effects of the force from the heart anymore, which is why you don't see this pressure coming back up to what it was before. So what we now rely on to get this blood back to the heart is less the pressure from the heart to begin with, and more the pressure that's exerted by muscles that surround some of these veins that lead back to the heart. So whenever the body moves, like say you walk or you stand up or that sort of thing, that muscle contraction that you do is also going to exert pressure on those veins, helping to drive the blood fluid upward back to the heart. And so this is why sometimes people will feel faint if they've been on their feet for a long time and they need to sit or lay down. It's also why, say, for example, a foot will fall asleep over time. It could be because you're having so much blood pooled in that one location that we need to sort of move a little bit to get that blood out of there, to resume normal nerve function, and to replenish that blood with new oxygen-rich blood. All right, so let's talk a little bit about how blood pressure is measured. I'm sure you've had this done before at a checkup, maybe here on campus even, or you'll be doing it in laboratory for class this week. So what happens here is they put on the cuff, and that cuff is going to be tightened using either, it can be electronic, or you can use a bulb that does this by hand. And you tighten this enough to cut off blood flow to the arm. So what you're doing is you're squeezing so hard on this artery right now that the blood is pooled right here, but it can't get through. So this part of the arm is not going to be feeling that blood flow. And so that's why sometimes you'll feel a little bit of a painful sensation down in here. It's only momentary, but it's because these cells are kind of deprived of oxygen. So usually they have to exert pressure above 120 psi. And so then what happens is what they then do is they relax this. And so during this time, blood is able to resume flow through here, and they're going to start paying attention to two different sounds. When the heart rate reappears, which is what's going on here, because at this point, if you put the stethoscope on the underside of this, there's no blood flow coming through here. So you should not be able to detect a pulse during this time. Over here, though, we're going to listen for the pressure at which that heartbeat resumes, because there's so much blood now flowing through this artery that it's very loud and you can hear it through the stethoscope. And then we're going to listen for when the sound goes away through the stethoscope, because you've basically resumed normal operations. It's back to baseline, that sort of thing. And so that's what the top number refers to when you get your blood pressure checked, and the bottom number. And so this has to do with the vibration and the diameter of your blood vessels during that time. All right, so blood pressure is an important indicator of heart disease. If you have heart disease, or if you have blood pressure within a particular range, that can indicate significant health problems. We have something called hypertension, which describes high blood pressure. And actually, we estimate that 1 in 3 American adults have hypertension. When you have this, what this means is that the diastolic pressure when your heart is contracting is going to remain abnormally high. And so what this means is that the heart has to work extra hard, and that can lead to enlargement of that left ventricle. Eventually, this can weaken the heart, and when the coronary blood supply especially when the coronary blood supply cannot accommodate that increase in muscle mass. And so as a downstream consequence, this also increases the pressure that's on those blood vessels that are immediately off the heart, things like the aorta, because over time, this stronger pressure because of an enlarged left ventricle can lead to tiny ruptures in that aorta, and you could even get something like aortic rupture. So, let's go back. Oh no, this is fine. We can stay there. So if you have hypertension, sometimes you can have this just because you were walking into the clinic, or you had coffee recently, or that sort of thing. That's okay. And for the most part, they'll pay attention and they'll try to get you to relax a little bit, which is part of the reason why you have to wait in a waiting room for so long. Before they measure your heart rate, they'll make sure that you don't have your legs crossed, because that can impede blood flow, et cetera, et cetera. Then they'll measure your blood pressure. But they're looking for a trend over time. If your blood pressure is elevated one day, that could be because of anxiety or stress, but they're going to monitor that over the long term to see if there is a trend that might indicate a problem. If you have prolonged hypertension, that is a major cause of heart attack, heart disease, stroke, as well as kidney failure. So we have 3 different stages that we describe when it relates to hypertension. If you have pre-hypertension, that means that your blood pressure, your systolic is 120 to 139, and your diastolic is 80 to 89. In stage 1 hypertension, then it's 140 to 159 for the systolic, and then for stage 2 hypertension, that's when your top number of your blood pressure is 160 or higher. But ideally, you want to be less than 120 and less than 80, so 120 over 80 or less. So things you can do to help your blood pressure is to observe a heart healthy diet. So eating lots of fruits and vegetables, watching things like sodium intake, just trying to observe good eating practices. It's also a risk factor to be a smoker, so that would be something you'd want to avoid. Avoid excess alcohol, limiting yourself to, and that would mean more than 2 drinks per day, so that's what you'd want to avoid. You're a college student, so I understand this is going to be a concern for some. And then, maintaining regular exercise to help maintain a healthy body weight can also help prevent heart disease. So now that we've talked a little bit about how blood moves throughout the body, I want to spend just a little second again talking about the exchange between the capillaries and interstitial fluid. Again, if we're looking here at an artery end of a capillary and the venous end of a capillary, remembering that these cells are just one cell, or these vessels are just one cell layer thick, and the interstitial fluid is out here, we've got two things going on here. We've got simple diffusion that's going to help move the nutrients and the gases out of the blood vessels into the tissues or vice versa. We're also going to have things in the tissue cells here like endocytosis and exocytosis that help to move larger molecules. But these capillaries are really leaky. They're, that you can have movement through these little gaps. And so these perforations help with the transport of things like water, salt, and sugar. Now, blood pressure that we've been talking about is one of those things that encourages this leakiness. We're going to have blood pressure that is exerting force outward from the capillaries. And then we're going to have osmotic pressure that is going to be pushing fluid back into the capillary, because the solute concentration in the capillary in here is greater than what's here in the interstitial fluid. So because it's higher here, the water is going to want to move into this space to equalize the concentrations. So when blood pressure is greater than, and because blood pressure is greater than osmotic pressure, this means that you're going to get a net loss of fluid over time from the capillary. And so we rely on a second system called your lymphatic system to collect any of that excess fluid and return it back to the heart and back to the body. So that returns again through the lymphatic system, and that is sort of a companion highway that overlaps in many ways with the circulatory system, but it's the highway of your immune system. And it's also going to include tiny networks of vessels that are mixed with those capillaries. A lot of them are going to be absorbing that net loss of fluid from the capillaries and returning that to the heart. It merges with your circulatory system in your neck. So that's where the interplay piece is. And this works together with the circulatory system to try to maintain fluid balance and hydration for the entire organism. So let's talk for a moment about blood. When you have a blood sample and you send it to the laboratory, one of the first things they're going to do is centrifuge it, which is kind of like putting a blood tube in, what do they call that? Not the tilt-a-whirl, the gravitron if you go to a carnival or a fair. It's that ride where they spin really, really fast, and that eventually kind of makes your face glue backwards into your head, that kind of thing. But what that does is it separates things by size. So lighter weight things are going to naturally appear at the top of the tube, whereas heavier things are going to be concentrated down at the bottom. So in the upper part of this sample, you'll notice in here that it's kind of a yellow or clearish sort of fluid. And that's because it's mostly water. This is called plasma. And this is 55% of your total blood makeup. So it's mostly water, but it's also going to have ions in it. It's going to have factors in your plasma that are important for things like clotting, so that you don't bleed out from a minor injury. Those sorts of things. It's also going to contain your antibodies, which are critical to your immune system, that sort of thing. And then any sort of substance that's transmitted or transported through the blood, but not necessarily inside of a cell. So this would include things like waste products, gases, hormones, those sorts of things. Glucose is going to be in the plasma. Then down in the bottom part, you have the heavier stuff. And so these would be cells mostly. And so we're going to have your white blood cells, which are an important part of your immune system. So that could include, lymphocytes and eosinophils, and et cetera, et cetera. We will also have platelets, which are important for blood clotting. They work with things like the fibrinogen that you see over here to form clots so that you can wall off an infection or wall off a capillary if it's been disrupted because of an injury, for example, like you scraped your knee, so that you don't bleed out. And then you're going to have red blood cells. And so this is the big one. You have 5 to 6 million of these, and their role is really the transport of oxygen and some CO2. You have then the number 2 thing you find in the blood the cellular debris or cellular elements are the platelets, and then rounding that out is the white blood cells. Now, if you have an infection, the proportion may change and you might have an elevated white blood cell count because you are fighting an infection. All right, this kind of summarizes what I just told you. So we'll just talk about some of the highlights here. Those monocytes and neutrophils, so that would be this one and this one, those are called phagocytes. So what they do is they swallow other cells and especially bacteria or other threats to the body. They will mostly spend their time in the lymphatic system and the interstitial fluid because especially the interstitial fluid is where you might find the bacteria or pathogens. Your red blood cells have no role whatsoever in the immune system. Their job exclusively is to carry oxygen. They look like slightly filled in donuts. They don't have a nucleus. They have lots of surface area to encourage oxygen diffusion. They have no nucleus, like I said, and that just gives them more space to carry hemoglobin. We estimate that they carry about 250 million molecules per cell, so lots of hemoglobin, and each hemoglobin molecule can carry 4 molecules of oxygen. The red blood cells, have a lifespan of about 3 to 4 months. They're made in the bone marrow of your body and at a rate of 2 million per second. So you're making a lot of these. These turn over time, and all of this rate depends on negative feedback from the kidneys. What they are doing is sensing how much oxygen is reaching the tissues. If the kidneys decide that we need more oxygen to reach the tissues, then they will secrete a hormone called erythropoietin. And what that does is stimulate greater red blood cell production in the bone marrow. You have an ideal range for this. If you have too few red blood cells, you have what's called anemia. Those kinds of individuals will usually experience things like constant fatigue because they don't have enough oxygen for their cells. And because oxygen is a critical element to getting ATP from glucose, if you don't have a lot of oxygen, you don't have a lot of ATP. If you don't have a lot of ATP, you're going to feel like you have low energy. There are causes for this anemia to include things like excessive blood loss, if you're hemorrhaging internally, for example, if you have certain vitamin or mineral deficiencies, people who have cancer, and women who are menstruating. Because they are bleeding, they would have, and they're losing that blood, that is considered an anemic time. So this is something you would more commonly find in women than in men. And so while we're talking about red blood cells, and we're talking about erythropoietin, it's important to talk about the fact that you have certain adaptations that you make as a human, whether you're operating at sea level versus whether you're at altitude. So in high altitude conditions, like what Maria von Trapp here is telling us about the hills being alive in Austria in The Sound of Music, is that because oxygen is low, we need more red blood cells to gather up as much oxygen as we can because it's in short supply. And so for that reason, this management of red blood cells is something, because the red blood cells are carrying oxygen and that directly relates to how much energy you can produce. This is one of those things that endurance athletes like to try to control and manage. And so what you'll find is that for a lot of professional athletes, and especially endurance athletes, they may train at altitude. And that's why a lot of our U.S. Olympic training centers are in places like Colorado, is they will train for a month or so at altitude, and then they will go compete at sea level. When they do so, they have more red blood cells circulating in their blood, because remember, these things turn over every, 3 to 4 months. Then they will have a higher red blood cell count than somebody who is training at sea level. But that erythropoietin hormone that we were talking about that the kidneys produce, there have been situations in which people have isolated EPO, or they have created synthetic erythropoietin and used it for blood doping. Because rather than training at altitude, then we could just give the person this hormone and that would stimulate additional red blood cell production without them having to train at altitude. Or it could confer an even bigger advantage to those who are training at altitude. So what happens here is you extract and then you store a person's red blood cells, blood doping can feature synthetic EPO. It can also feature when you extract and store somebody's red blood cells only to inject them again just prior to competition. Because again, that increases their ability to get oxygen from their environment and then deliver that oxygen to the cells so that they can do work, so that those muscles can continue to work over a prolonged period of time. But while this sounds like a good idea on paper for endurance athletes, this can be fatal if an athlete happens to get dehydrated, because you need a certain amount of water in order to do in order to carry all of these red blood cells through the blood vessels. And so we've talked about why this would help in competition, but because of the risks, blood doping is something that is banned and illegal in professional athleticism, in the Olympics, those sorts of things. And two of our most prominent examples of people who have been caught doing this include Lance Armstrong, who was a 6-time Tour de France champion. The asterisk is there because he was then, those titles were stripped away from him when they figured out that it turned out he was cheating and using EPO this whole time. He's been banned from life from competition from the Tour de France. He has since gone into like triathlon and some other things, but I don't think that he's ever going to be able to be considered a professional athlete again in that sense. Another example is Marion Jones. She was a 3-time gold medalist in track and field at the Sydney Olympics. She received 6 months in federal prison, not because of the doping, but because of something having to do with lying to investigators. She wrote some bad checks. There were some other things that happened, but this was tied up in the Balco scandal, which also tied up Barry Bonds, a baseball player from the San Francisco Giants. All right, so let's talk for a moment about blood clots. I talked about how you use platelets and you use fibrinogen to form these clots. Let's say that you're wounded. You fall off your bike and you scrape your leg. Well, we don't want you to bleed to death from something relatively minor like that. And so you have these self-sealing materials, these platelets, this fibrinogen that can be activated if you have a blood vessel that's damaged. And so what happens first, let's say there's an injury. Here's the injury right here. What's going to happen is you're going to constrict blood flow to that, or constrict that blood vessel to limit the amount of blood, that's flowing through there and could potentially leak out. And this also allows time for repair. So what's going to happen is the platelets, which are not activated yet, are going to bind and recognize a site of injury where the epithelium and connective tissue have disconnected from one another. They will then release chemicals that help to make other platelets in the area become very, very sticky. And so they form what's called a platelet plug. What then happens is that plug is going to limit the amount of blood loss through this opening, and eventually you build a clot with the use of fibrin that helps to act like a scab inside of the blood vessel. So that's shown here in white. So this is a clot, and you can see that it's going to occlude the movement of red blood cells through this space. But during this time, we have healing that's taking place. If you don't have enough clotting, or you're not able to clot, that's called hemophilia. And this is that thing that we talked about where Queen Victoria was the unknown carrier for this gene and then spread it throughout the royal houses of Europe. So these people can die from a seemingly minor injury because they can't form a scab, they can't form a clot. You can also have a problem where you form too much clotting, or the clotting is happening inappropriately. This is called a thrombus when this happens. If it happens within a blood vessel, that's called a blockage. And if that blockage takes place impeding the blood flow to the heart, then that can trigger a heart attack. If the clot impedes blood flow to the brain, then that can cause a stroke. But this can also happen in the eye. And when that happens, you develop sudden blindness, which may or may not be reversible. So that leads us to the review slides that support this lecture. I encourage you to go through these things to make sure that you understand more fully the concepts that we talked about, some of the vocabulary terms, that sort of thing. There's also a diagram on the last page. We went through this before in lecture, but see if you can figure out how to label this structure. You're going to get practice doing that here in lecture, but you'll also get practice doing this in lab. So this is in your review packet, but it's also provided to you here. The other thing I wanted to talk about is for your participation points, to get you the points that would normally be awarded for today, you need to go to D2L. You need to go to Unit 4. And then you need to go, I know we just covered circulatory system, but you need to go to gas exchange. And there are two things you need to do here. There is a video for a TED Ed talk that summarizes how oxygen moves throughout the body. And it's a good summary of what we've talked about so far in terms of the interplay between the respiratory system and the circulatory system. You need to download and print or otherwise copy and paste this into an electronic document that you can submit to me via email. But this is just a multiple choice sort of handout that you're doing as a companion to that TED Ed video. This will be due at 8 o'clock or 2 o'clock, depending on your section, but at the start of our next class period. That's when it will be due for the 5 points that would be awarded for this day that we're having to miss in lecture. So I think that concludes what we were doing here, and thank you very much.